Good afternoon, everyone. Today, I will present my seminar under the title Morphological and Molecular Diversity of Leucocoridium, Parasiticizing the Semi-slug family Saxinidae, case study in Japan, Russia, and Argentina. The Leucocoridium is the part of the class Trematoda, a group of parasitic flatworm commonly known as fluke. In this picture is the zombie snail. In the wild, zombie also exists. Like in this picture, the amber snail, which became a zombie snail after being infected by the trematode Leucocoridium. The Leucocoridium has multiple hosts. In this picture, after infection, amber snail turn into the zombie snail with larval brood sac. Pulsing and colorful seem to be the caterpillar mimic for attracting the insectivore bird. The trematode transfer in the new host after the bird eat the brood sac, grow up, and reproduce in the bird's intestine. Leucocoridium release its eggs with bird feces and transfer into the amber snail again. The different state of the amber snail occupy different hosts and host organ. The gravid female gravid adult live in the bird cloaca and release the egg into the environment within the bird feces. After that, the amber snail eat the feces with the egg and turn into the multitubular sporocyte that grow from the hepatopancreas into the body cavity. Asexual multiplication of the cercaria occur in the sporocyte, which continuously develop into encysted metacercaria. A fully developed sporocyte, namely mature brood sac, exhibit a rhythmic activity in the eye stalk. Now, Leucoridium has been reported in 7 species around the world. However, they still contain the taxonomic problems for the species identification worldwide too. That brings me to the question of this work, like what are the species of diversity and distribution of the Leucocoridium in Japan? And what is the population genetics of the Leucocoridium paradoxum in Europe? What is the Leucocoridium diversity in Southern America and the evolutionary relationship of the world Leucocoridium? The objective of this work are to clarify the taxonomy, distribution, and species diversity of Leucocoridium in Japan, and to analyze the genetic variation of L. paradoxum sporosis in Europe, and to explore Leucocoridium diversity in the Southern America and analyze the evolutionary relationship of the world Leucocoridium. The sampling site include 3 different parts of the world, namely Argentina, Russia, and Japan. The sampling include sampling the Leucocoridium as different states from the Saxinia snail, such as brood sac and metacercaria. The adult worm was sampled from the insectivore bird, feces, and cloaca. After that, the sample will do the DNA extraction and the gene in this study including CO1 gene, 28S RNA, and ITS gene. The result in Japan, 3 forms of the brood sac were found. These 3 species have the distinct different characteristics, which the host of them are the alien snail from North America. So you can see the different coloration of the brood sac, including the brown band brood sac in Hokkaido and green band brood sac in Hokkaido too. And this one, the vertical brood sac that found only in the Okinawa. Here show the distribution of these in Japan. Leucochoridium species were observed in Hokkaido, Honshu, and Okinawa. Most of them were in Hokkaido. The larvae and adult were previously recorded in Honshu and Hokkaido. So in Hokkaido contain 2 species, the brown band and the green band, and the Okinawa contain vertical strip and green band. Here is the result of the 28S RNA, which is unusable for the microtaxonomy of the congeneric species due to the low resolution. The data from the COX-1 and ITS show that the L. paradoxum in Japan is the same class of the L. paradoxum in Europe. And the unidentified Leucochoridium found in this work are closely related to the L. Voctinium in Europe too. The haplotype network of L. perturbatum and L. paradoxum reveal that L. perturbatum is more diverse than the L. paradoxum. So in this work, Leucochoridium that have strip vertical in Okinawa pattern show the similar to the L. passeri in Taiwan, and molecular results show that Leucochoridium in Japan similar to the Leucochoridium voctinum in Europe. However, this unidentified still cannot be determined because they infect in a different host, and only 2 species that can be identified. Let's move to the Russian Leucochoridium. So in this picture, you can see the sporocyst of L. paradoxum that show the difference of maturity by color. The most mature brood sac has intense pigmentation that bear an olive brown strip, and the sporocyst of the Leucochoridium that have the variation in their color. All are infect the intermediate host Saxinia putris. The sampling were conducted in Russia and Belarus. And even the European brood sac color are vary, but they share 5 main characters, which are: first, start from the dark brown spot, followed by the upper band and the light zone, the lower band, and the proximal part with light yellow and green band together. So here, the analysis of our data and the sequence in the GenBank reveal 27 haplotypes of L paradoxum. So the most numerous was haplotype number 3, which are from the Japan. So the haplotype network divided into two groups, the European part in Eurasia and Japan sporoseed that different by 20 nucleotide substitution. L paradoxum and L perturbatum and L vaccinum. Should be noted that two main clades form within L paradoxum and is divided into the Japanese clade and European clade. So you can see the L paradoxum in Russia and Japan grouping in the different clade. The L paradoxum in Europe and Japan is similar, but they show no correlation of the color and the location. And however, the Europe and Japan haplotype are separated. The difference between the Japan and European group is 0.0324, that higher than intraspecific genetic distance in the family Leucochoridae, which range from 0.005 to 0.01. And Leucochoridium from Japan may belonging to the different species from the Europe. L paradoxum highly specific to the intermediate host, but less specific to the definite host. That's why the bird held the trematode on the migration into the different continents. And last one, the case in Argentina. The specimen of the host Omalonyx semislug were collected during the file sampling event from March 2022 to December 2022. And this snail is totally different from the previous two studies because they live in the aquatic plant. The host, the definitive host that observed in this sampling area was native Jacana and Vanellus chilensis, which also different from the previous two study too. The study conduct in Argentina, and this one showed the distribution map of the Leucochoridium in South America. They consist of the green band, brown band, and the root sac of green and brown band. So in this work, a total of 140 snails were collected between March and December 2020, with the only one that found to be the infected. And this brood sac, the color dark bar on the top here, and followed by the light yellow strip About the adult, they mostly found in the bird passeriformes and focus on the dissecting the cloaca of the birds. You can see the uterine duct that running parallel to the thoracic and both draining into the genital atrium. With compare with the L paradoxum, it can be seen that the position of the uterine duct is different from the thoracic. So the leucocoridium that found in this work is closely related to the L paradoxum in Russia and L perturbatum in Europe. The mitochondrial marker from the maximum likelihood and Bayesian reveal the similar grouping but show some different topography. And Leucocoridium sp from Argentina consistently cluster with L perturbatum with a highly support of 99%. The species delimitation analysis result in 7 or 8 putative species of the C1 dataset, with the number vary depending on the method. And they recognize the Leucocoridium from the Argentina as a distinct species. So the discussion, this study is the first molecular work of Leucocoridium in America and focus on only the brown band brood sac and metasaccharide found in Omanix and goose. The molecular and morphological analysis confirm the distinctive of the Leucocoridium in Argentina. The characteristic of the brood sac coloration pattern and a good indicator of the phylogenetic difference. The study recommend the further research with more genetic data to clarify overall the evolutionary relationship between the world Leucocoridium. So, the conclusion of this work, in Japan, 3 species of the Leucocoridium were observed and 2 of them can be identified. But in the Europe, the Leucocoridium paradoxum was analyzed and the species was confirmed, while the Argentina, 1 species of the Leucocoridium were found, but still unable to confirm. This one is the selected reference of this work. Well, thank you for your attention, and please feel free to ask any question.